Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. At 7, we'll see you there.
Tell me a little bit what you can really translate from the yeast now to your models, applying that to metabolism and maybe even in humans. Where are you at the stage that we can really have the digital twin?
The machine learning part, that one I could say it's a simple because that's like a methodology that it's already developed, and we already take a lot of health data and analyze it. And there are a lot of papers coming daily, basically. But the core here is this mechanistic, when you derive the mechanism, why this is happening. And this is important for the predicting outcome. And we are now at the stage where we have a metabolic model of human. That is one module that it's ready.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us. You just saw a short piece of our last episode with Professor Maria Priotkic on understanding aging through computational modeling. And you can see the full episode on YouTube. Today, we talk about biomarker profiling. Professor Karsten Weylandt is with us tonight. Routine assessments for biological age determination might trigger many questions, at least in me. Please use the Q&A function to send your questions, suggestions, whatever you want to share with us. Delphine Chen, a PhD student, will moderate your questions today, and she will introduce herself first.
Nice to meet you. Could you tell me a little bit about yourself?
Hi, my name is Delphine. I'm a third-year PhD candidate currently working closely with Dr. Vivian from the Department of Nursing, who is under the Healthy Longevity Translational Research Program.
Well, that's so cool. You're probably our first member from the nursing department. Could you tell me what about nursing that interests you?
Well, I think nursing is both an art and a science. For nursing, we really look at healing not just the physical body, but the heart as well. So what inspired me to do nursing is that earlier in my life, I volunteered in the acute hospital to actually give out magazines to patients who are warded within the hospital. I interacted with a lot of patients, and the patients actually show a lot of appreciation for the kind of things that we are doing. And I find that being able to take care of patients when they are most vulnerable is something that my heart really feels aligned with, and that's why I joined nursing. Yeah.
That's so heartwarming. Thank you so much for sharing. What made you decide to do research then?
Through my experience working in the clinical, I realized that we need to base our care on evidence-based practices, and I find that there are many things that we could explore through research initiatives, questions that we can answer to help better patient care through participating in research activities. And I find that it is also time for me to actually move on to understand the science behind the kind of interventions that we are doing, and to create something that we can implement clinically to help to better the health outcomes of these patients.
So what's your current research about then?
My current research is on a multi-modal intervention looking at frailty and cognitive impairment. So we use cognitive as well as exercise, in-person training and virtual training to help to mitigate the risk of frailty and cognitive impairment within the community dwelling older adults in the active aging settings.
What is your favorite moment working with participants?
I would say that it's during the cognitive training where we had a lot of fun.
What's the thing? I hear black, green, red. Three colors. Green. Green. Final answer.
is something that is mentally straining for the participants. But during the cognitive training, we actually interacted a lot and have a lot of fun moments where we realize that there's many things that we taken for granted for our cognitive abilities. So through that, we actually learn a lot about not just how to improve our cognitive abilities, but also to learn how to motivate each other in that cognitive training process. And I've seen the seniors growing from a group of strangers into a group of friends who are supporting each other through this journey. It really warms my heart and makes it the best moment for me during the intervention.
That's such a heartwarming initiative. Thank you so much for sharing that. So how has the Healthy Longevity Translational Research Program changed you or inspired you?
The Healthy Longevity Translational Research Program helps me to get in touch with people from other faculty other than just my own faculty, and I learned that there's a lot of different branches of science that I previously wasn't so aware of. And I also managed to join the Young Scientist Club that was an initiative by the HLTRP, and this has helped me to form many beautiful friendships on top of the research that we are doing and the activities that we are doing to promote the young scientists within the community. And it also helps us to actually have a lot of opportunity to interact with people from other faculty and discover more research, collaboration and opportunities.
That's so sweet. My last question is, what do you enjoy doing outside of work during your free time?
During my free time, I enjoy playing with my pets at home. I have a dog as well as a munchkin cat whom are both adopted. So just a fun fact, I think the munchkin cat came to our house 2 years ago unannounced, and we actually went through a lot to bring the munchkin cat back into our house as our pet. And I think he has added a lot of life into our family, and now it's the best companion of my dog. And other than that, I also do community grassroots events at the Yio Chu Kang community to actually help out the residents in learning more about the community itself and also to bond as a group. We created a lot of activities that will help us to improve the welfare of the residents within the community.
That is so sweet. Thank you so much for sharing everything with me, Delphine. How can we learn more about you?
You can find me in LinkedIn or email, and I will also be moderating the session later. Please feel free to leave your comments in the chat box below.
Thank you.
Yeah.
Bye.
Thank you. Bye.
Thank you, Delphine. There are so many joyful events, I think, in your life. And I love the cat story, being unannounced, coming and knocking on your door. Good. So we are going from lovely stories of how to prevent frailty and cognitive decline with very difficult tests, now towards biomarkers of aging, of health. And our guest today is Professor Karsten Weylandt. He is a gastroenterologist at the Brandenburg Medical School with a background that spans clinical medicine, molecular biology, and nutrition. He works as focusing on fatty acids, lipid metabolism, and metabolic diseases with a strong interest, how pathways influence aging, and how it can be translated into healthy longevity medicine, which is, of course, really our topic. So, Karsten, thank you so much for joining. We would love to hear how we can estimate biological age very easily in clinical practice.
Well, thank you very much, Andrea. Delphine, thank you for the invitation. Thank you for being able to discuss with you, because I'm not sure about what this actually implies, what we are measuring, but I've started sort of being interested in that. You said you told everybody already that I'm a gastroenterologist, so my work is endoscopy in a lot of contexts, and there we do sort of longevity by preventing colon cancer mostly, right? So this is probably what we kind of try. Well, this is how we try to prevent disease. We probably come more from this prevention background when looking at that. So I was more thinking about prevention. I'll tell you a little bit about how far back this goes with what I've been doing. But the new thing that has added quite some fun to my life doing the clinic and dealing with our walk-in patients has been to sort of take into account this marker-based, or to start think about how to measure age or biological age, or how to measure risk differently from what we've been doing on a more routine basis. And that's what I would like to sort of introduce to you, and then we can and I hope we can. I have more questions probably than answers, much more questions actually. But I hope we can hopefully go into that in the discussion a little bit. But first of all, let me show you a little bit how I actually started thinking about this kind of stuff. And I don't think that professionally about it, so I'm more of an amateur in longevity medicine, I must admit. So thank you very much that I can actually join you today, and I can share some of the stuff we have been looking at, or we have started doing. So now I would like to let me just share my presentation, and then we can start into that. Okay, so my topic shall be biological age estimation in the clinic using routine biomarkers. Maybe I should have put the title a little bit differently and should have written in the routine clinic using biomarkers. This is basically our setting. It's a hospital right outside of Berlin in Brandenburg, and this is where we are. It has been a psychiatric hospital 100 years ago. Now it's sort of a general hospital, and this is where we have a gastroenterology department and a large outpatient department. And as aging is not a disease, we are not treating aging. We're treating GI diseases. We are doing endoscopy to prevent things or to diagnose things, and we do metabolic medicine to alleviate metabolic risk. How I stumbled into this field of longevity and of measuring age was through an let's see how it works there. Through an article in a German general newspaper that I read when I was flying to the Canary Islands a few years ago. I put this here in German because that's how I got into it. Researchers claim they have conquered aging, right? And this is about putting the clock, the aging clock, backwards and rejuvenating people. And this is this newspaper article was about this study by and all of you know that study much better than I do, by Greg Fahy and Steve Horvath, where they show in a small set of probands that by reversing by using a combination of growth hormone and DHEA and metformin, that they were able to turn the clock backwards, which got me into that context, into this idea to actually look at age differently. I didn't think about that very much before that. And so I got into the whole field of measuring age, and it was through Jan Gruber and Feng Sheng in Singapore that I really started to get an idea and to grasp the idea of clinical aging clocks, of how we can actually use parameters. I read about the methylation clocks, Steve Horvath and people, but looking at biomarker-based clocks, Jan really put me into that and got me the idea and got me the understanding. And my background is more in inflammatory medicine and in oncology. So I was looking at totally different contexts. I wasn't looking at how to measure biological age versus chronological age to actually understand aging. And Jan got me into this paradigm of thinking. And I realized that it's very close to, actually, in a way, very close to what happened in cancer medicine in the last 100 years, maybe. And I have a quotation here from Siddhartha Mukherjee, the emperor of all maladies on cancer, about how Faber in the 40s and 50s started looking at cancer medicine. And he writes there, if cancer medicine was to be transformed into a rigorous science, then cancer would need to be counted somehow, measured in some reliable, reproducible way. And Faber started with leukemia because he was able to measure blood cells and to measure leukemia in the blood. And at that time, there were no CT or MRI scans, so there was not a possibility to measure solid tumors. But this is the basic idea, and this is, I think, the basic idea of why I think, or why I find it fascinating to think about biological age measurements in clinical medicine, in routine medicine, because if we want to understand a phenomenon and scientifically, and if we want then to be able to actually intervene and to test interventions, we must first be able to describe it well, and to describe it well, we must be able to measure it. And this is how I arrived at trying to measure age beyond the idea of chronological age as a likelihood of dying irrespective of chronological age or with relation to other contexts. And this is how I then, because I'm in a routine clinic, I talked to Jan about the lineage parameters that Fengsheng and Jan established in their paper, and I found that not very realistic for our setting of routine clinics to use this broad parameter spectrum of biomarkers in a routine setting. So that was when I stumbled upon the pheno-age biomarkers that were established in this paper by Morgan Levine and Steve Horvath in 2018, based on enhanced data, establishing a small set of biomarkers that they used to calculate a phenotypical age from this cohort of patients. And this biomarker-based aging clock is pretty simple, pretty straightforward, and based on a small set of parameters that we can actually, even in a routine clinical setting in Germany, where we have where we see patients on mandatory health insurance, where we don't have any specific research money to go beyond big biomarker panel measurements for the routine patients. This was something I thought was possibly feasible to do in our little clinic. What I want to show you now on the next few slides is what we actually, so it's the main paradigm that we pursue in our metabolic clinic, and that is sort of very classical cardiovascular medicine. Yeah. When you want to imagine what we do in our with our patients, we focus very much on, and that has also to do with my training in lipidology. We focus very much on LDL cholesterol as risk factor. And you see here data from a paper from 2017 showing how the LDL cholesterol, the low density lipoprotein cholesterol, actually associates with atherosclerotic lesions, even hidden or subclinical atherosclerotic lesions, depending on the level of the LDL cholesterol. So when you look here, you see even if you don't have any clinical manifestation of atherosclerosis, with higher levels of LDL cholesterol, you can see atherosclerosis in these patients. So this is the paradigm that we, from this metabolic cardiovascular point of view, use in our clinic, and I like this figure very much from Eugene Brownwell's editorial in the European Heart Journal in 2022, where he argues that the cumulative LDL-C burden is the factor actually dictating when atherosclerotic disease becomes clinically relevant and prevalent, and where he argues that by sort of lowering LDL cholesterol and lowering the slope of these of this accumulation of LDL cholesterol in the human body, we can extend atherosclerosis-free lifespan. He argues we can do that with inclisiran, which is a very, very cool substance where it's an siRNA to lower LDL cholesterol, and you only have to inject it twice per year. And he argues you could probably do something like giving an injection once a year, like the flu shot, to lower LDL cholesterol, and to by that achieve long-term atherosclerosis-free or atherosclerosis poor, low survival. So this is our little prevention paradigm that we that we use when we treat our patients in our metabolic clinic, and that's based on the treatment goals defined by the European Society for Cardiology. How you treat and how rigorous you treat LDL cholesterol levels depending on the background of disease levels the patient has. So you have someone who has a low score, low risk of atherosclerotic events, then you can be a little bit more relaxed on for the value for the target value of the LDL cholesterol. And if you have someone who's very, very sick and who has already a lot of atherosclerotic disease, you have to treat those patients much more intensely to actually lower LDL cholesterol to say 1.4 millimole per liter in the high risk group. And so when you what I show you now on the next two slides is what we actually saw in our patients, and I had a first small data set that I showed that I had on a poster at ARDD this summer, where we were able to where we did a 1 year follow-up of patients of 54 patients from our clinic, and we were able to see that this is a little result group. We come to a bigger result group in a second. That by lowering cholesterol, that in our clinic we were able to lower cholesterol. Here there was the lowering was only significant for the total cholesterol, not for LDL cholesterol. But in the bigger group, and that is from approximately 640 patients, we were able to see over after a mean follow-up of 265 days, we were able to see highly significant lowering, not extensive, but highly significant lowering of LDL cholesterol and cholesterol in this group of patients. And these are the patients that at the same time when we targeted, and that is what we did, what we do as an intervention, this is kind of what we are geared on in our clinic. These are the patients we also measured using the PhenoAge biomarker parameters. And what we saw there was, and that was kind of a cool thing with the first data set, and I had that sort of hidden on the poster because I couldn't really believe it that that is totally true, and it isn't totally true. When we looked at the PhenoAge calculated from these PhenoAge biomarkers in the small data set of 55 patients, we were able to see that over a follow-up of 323 days, so close to 1 year, their chronological age increased by a year. But at the same time, in these 54 patients, the PhenoAge measured with this little set of biomarkers actually decreased. So we made those 54 patients younger, over that year, looking at biomarker-based PhenoAge. I couldn't, this is really too good to be true and a too small number of patients to look at. So for today, I looked at, so it's December, I can now look at basically the whole of 2025 in our data and can compare 24, 25. So what I did the last few days to show you what happens in our bigger patient group, when you look at our PhenoAge biomarkers there, we see we have a follow-up of 8.7 months, not a year. And obviously chronological age increases by these 8.7 months. And in this bigger group of patients, we had a phenotypical age that was calculated higher than the chronological age. But what we saw with our LDL cholesterol geared intervention, this is kind of our target size, more or less. We say eat more healthily, but more or less our main focus is optimize LDL cholesterol control. We were able to see that that in this group of patients, the increase in PhenoAge slowed as compared to the chronological age. And when you look at our lipid biomarkers in this group of patients, you can see that this big group, we were able to see what I showed you before. We were able to see that we lowered LDL cholesterol. We didn't change HbA1c though. And so what we see here, apparently, when we look at phenotypical age, we look at parameters that are not, we change parameters that are not part of the calculation of the PhenoAge, of the biomarker-based PhenoAge, because there you don't have anything from the lipid parameters as part of the biomarker data set that is used to calculate this phenotypical age calculation, phenotypical age estimation. Part of it is the metabolic parameter, the glucose. So the HbA1c, as a sort of derivative of the glucose, is part of the pheno-age calculation, but not the LDL cholesterol, not the triglycerides, not the total cholesterol. So from that, and I hope we can discuss that once I've finished the second part, which I'm kind of brushed through a little bit now, maybe. From that, it seems that we can be comfortable that we're doing something good for our patients if this pheno-age, biomarker pheno-age is something we can trust. But can we trust? That's something that I cannot really judge. But it's something I would like to explore further with you. Okay, so this is sort of the mainstay and the routine mainstay and the established mainstay of what we do in the clinic, because LDL cholesterol, I showed you the guidelines. There are defined levels. It's a very established, very well-established risk parameter. We know from animal experiments, we know from human studies, you lower LDL cholesterol, you lower atherosclerosis risk. And what we see is we do that, and at the same time we apparently decrease age acceleration in pheno-age. The second topic that we deal with a lot is sort of an old interest that I have. It deals with omega-3 fatty acids. You might not be that familiar with these fatty acids. I wasn't until I was 21 or so, when I started to work on that, and I show you how I started and how I stumbled into that. So there are essential long-chain fatty acids that we have to take up with our food. Those are the omega-6 and omega-3 polyunsaturated fatty acids, arachidonic acid, the paradigmatic omega-6 fatty acids, icosapentaenoic acid, and docosahexaenoic acid, the paradigmatic omega-3 fatty acids. And I got into this when I was a young medical student in Germany, when I was able, I had the privilege to join Alexander Leaf in his lab in Boston. He was at the time already a retired professor of medicine at Mass General Hospital, and my very first paper was from his lab, and was a small study on tissue cultures on primary heart cells, where we looked at the effect of essential fatty acids on spontaneously beating cardiomyocytes. I didn't realize at the time, and I kind of revisited that the last few years a little bit more when I started thinking more about measuring age and healthy longevity and lifespan extension, that Dr. Leaf was part of that thinking for quite a while, even before most of us here were probably born. This is a citation from a quotation from the obituary from 2013, when Dr. Leaf died at 92, and he had actually told me about these visits he did in the 70s, when he undertook a series of expeditions to study longevity in parts of the world where heart disease was rare and some people were said to live 140 years or more. And so he was sent to several places by the National Geographic Society to interview people and to ask them about their age. It turned out that a lot of that databases was a bit fragile, as many of these people were not really 140, but there was more a sort of a cultural thing that the origin date of birth was not noted, and everybody who was very old was said to be 100 or 120 or so. It wasn't really that he found the holy grail of going to 100 or 140, but his strong idea he took from these travels was that he believed in people living in mountainous places, working outdoors, consuming local food high in vegetable content and low in animal fat, that were the sort of the parameters defining healthy longevity. And that is what he took from these from these interviews, and that is what he took into his research in the Department of Preventive Medicine that he founded at Mass General Hospital, and where I had the privilege to join as a young medical student to do this first project. So I kind of talk too much about these kind of things and go more into the databases that is not so very strong for omega-3 fatty acids and cardiovascular prevention. There are these data from the JELIS study done in Japan already in the 1990s, where it was shown that with icosapentaenoic acid, with this one omega-3 fatty acids, you could, on top of the medication with statins to lower LDL cholesterol, you could reduce the rate of cardiovascular events. And I go into the next slides where I can sort of just walk you quickly through data that have been around for quite a while and have been published in many places, that omega-3 fatty acids apparently increase in age, or might be shown here, might be a marker of longevity. This study by William Harris, who established the omega-3 index, EPA plus DHA as a biomarker, showing that the patients or probants with the highest levels of these fatty acids had the lowest risk of premature death. We look at that as well. This is something beyond the routine lab parameters that I've been talking about so far. And when you look at in our little results from something like 600 patients in the past few years, we were able to see there as well that there was an increase in omega-3 fatty acids. Sorry, I just need to change this on. Sorry, I just had to change this on the computer. So we were able to see this increase in the omega-3 index in our patients, with patients that were older. We don't know whether that is an effect of diet, but it's an interesting finding because it's a very reproducible finding, and it's something that I think is worth exploring further, particularly since we know from this recently published study by Heike Bischoff-Ferrari from Basel and together with Steve Horvath, that supplementing omega-3 fatty acids was able to slow biological age acceleration, as shown in these data that you probably all know very well, and that were done in a much more complex setting than what we do in our clinic, where we look at these 8 biomarkers and not do any methylation clocks or any more complicated and more sophisticated clocks to actually measure biological age. And, I just wanted to sort of show you the broad spectrum that they did measuring this effect of omega-3 fatty acids. And what we did, we wanted to look in our cohort to at least to establish where the omega-3 fatty acids levels were in patients, depending on the quartile of their biological or phenotypical age, measured as pheno-age, biomarker pheno-age. Yeah, and so the last thing that I want to show you from our data, going quite away from the at first glance from the routine lipid parameters, I want to show you what we see when we look at omega-3 fatty acids in blood from our patients, close to 680 patients in total, which we separated into the quartiles. When we compare the pheno-age higher than chronological age, the highest quartile, so the oldest patients versus the youngest patients, we were able to show a small but significant increase or higher level of DHA in those patients that were phenotypically younger than chronologically. So the mean age of these patients was pretty similar in both of these, in the upper and the lower quartile. 62 in the old quartile, 59 in the youngest quartile, and mean increase, phenotypic age increase in the oldest group was 10 years, and the mean decrease in the lowest group was minus 7.5 years. Small difference, but something that I think we would like to follow up, and something I would like to discuss and think more about with you. What we also did in these in these patients, we obviously also measured all the other routine parameters that I showed you before. And what we saw here was that there was a big difference, a highly significant difference in HbA1c between these two groups. So the older patients had a significantly higher HbA1c, and so 3 stars here means many more zeros, really. And interestingly though, what we saw here was that the cholesterol and the LDL cholesterol was higher in the younger group, which at first glance is probably a bit confusing. And I would like to, and I'm a little bit confused by that. I might offer you a bit of an explanation in the next two slides, but it's something that I hope we can maybe discuss a little bit in the next minutes. So here we see the younger patients have higher, phenotypically younger patients have higher LDL cholesterol than the older ones, and they have a bit higher omega-3 fatty acids. So when we look at the PhenoH biomarkers, these 8 ones, we see that glucose obviously is part of it. Higher glucose means higher HbA1c. So this might be directly affect the PhenoH calculation. In contrast to these parameters that don't really affect the PhenoH calculation. On the other hand, though, when you look at these patients, you see that the older patients have higher triglyceride, they have higher HbA1c, so they are probably sicker. And when you look at this, what I also showed you before, is that the sicker the patients are, the more stringent and more rigorous are our approaches towards LDL cholesterol lowering. So something that I need to explore, obviously, I haven't done that for this analysis, right? Is whether this group of patients is just much better treated to lower LDL cholesterol than this group. And with this kind of probably a little bit confusing outlook, I would like to thank you for your attention, and I look forward to the discussion with you. And I hope I'm not too much over time. The big question that I ask myself all the time, looking at fatty acids since 1995, when I was in Dr. Leaf's lab for the first time, is whether we should eat more fat. And we should have done that the last 30 years. We would probably have less of an obesity problem worldwide. But that is maybe also an issue for the discussion. Thank you very much. And I stop my screen sharing, and I look forward to the discussion.
Thank you so much. Very interesting presentation. Let me start with asking you, do you give to your patients the PhenoH?
I don't. I wanted to do that in the beginning. And then I had a patient who was 62 and PhenoH was 95. And I thought, oh, maybe not a good idea. But that's something, yeah. What do you think? Should I?
So we, of course, develop these kind of algorithms. And at a certain point, we as clinicians have to discuss and convince ourselves, or being convinced by others, when is it ready for prime time in clinical practice? And you show convincing results with associations. We always have to ask ourselves, what is the consequence out of it? So how will a patient react? And we don't have yet much research, also from a psychological aspect, that if I would be 40, which I'm not, but I would be 60, if that would trigger depression.
I felt very good. I did it, and I was sort of 10 years younger than I am, which I liked very much. I don't know what I would have done if I would have been 10 years older or so. We are thinking, it's a very good question you're asking, how to deal with that, because we were discussing here in our clinic to actually, and that has so far we haven't managed to do that because IT people were not really up to. They said they were busy with doing other things, but we wanted to actually to establish to establish a module that automatically, whenever all these values are there, that PhenoH would be calculated automatically, and so we could take it into account when we have, for example, a patient with pancreatic cancer. And you have a patient with pancreatic cancer who's 60, but his PhenoH is 80. That might be something, even if it's the question is it enough evidence-based that we can use it as an additional parameter? Delphine, you look at frailty, right? Frailty is an important parameter. When you have a patient who's 60 and has got a pancreatic cancer and he is totally frail, much worse prognosis as compared, well, prognosis bad anyway, but much worse post-operative prognosis as compared to someone who's running a marathon. Yeah. But that's the big question, really. That's the holy grail in a way, right?
So I use biological age clocks in clinical practice, but not only one, but a plethora of it. So multiple, and sometimes it's very organ specific, sometimes it's more general. And I think what we have to learn in the field, that we have to distinguish between what is screening, what is assessment, and what is a diagnostic. And these age clock are absolutely not a diagnostic tool yet. I think everybody would agree. But the question is, can we use it as screening tools? And can we then further assess? Because what you are doing also in clinical practice, you are looking then at the sub factors within that score. Because then you're saying, okay, which of the factors, is it glucose or is it LDL, is actually driving that higher age? And I think clinically that is so meaningful.
Yeah, that's interesting. What is driving and how, and do we actually actionable consequence, right? Which is, I think it's quite, for us, it's quite neat that we have with the PhenoH something we are not actioning on any of it, maybe glucose a little bit, but we're actually putting all our action on LDL cholesterol mostly right now. And still we change PhenoH. This kind of, it makes it very interesting. This finding, looking at different biological clocks, that would be obviously something that we would like to do, and we have the samples, and I'm thinking about whether I have some money to do some kind of methylation clock on the samples that I have, and I have the ethics to do that, so we could do that. On the other hand, it's that is again the big question of the whole measurement issue per se, right? When we now end up, in the end, when we end up having tons of different clocks, then the whole beauty of the clock goes away. It would be so cool to have one or two, and then to be able, to say, okay, this is it, that is sort of what I'm dreaming of, really.
Think about your body of the different organ systems, because you also think that you not just have one body, but we are thinking about organ systems, and every organ system can age differently. So everybody has a tiny clock, and they sometimes have to be synchronized with certain therapeutics, and some need a little bit of attention there, and some other interventions somewhere else at the, of course, important time point. But let me ask the next question, following up with your great results, that actually the PhenoH was only increased by 4 months within that time period of 8 months, when you actually were showing that your LDL was lowered. People could say they were already biologically older in your bigger group compared to the chronological age. Is 4 months enough? Like, why are you not more aggressive? So what, for example, did the CRP go? The CRP is within lineage. Was that also lower? Or can you be more aggressive? And could we actually tell people to, hey, your goal is to be your at least same chronological age?
I haven't, I must admit, I haven't had the time to look at the data properly for that, because there will be subgroups where more, this was a very slight LDL cholesterol lowering. So there will be a group where we lowered much more aggressively. And so it will be interesting to see how the effect is in their group. That's totally right. This is something that just for the sake of time and people and yeah, the routine clinic ticks on, and this is my fun project on the side, and I've got a few students for that, but not a whole
Oh, no,
department.
it's fine. I think it shows the beauty, how much we can actually change in each of the bodies. And there's so much capacity. So sometimes we should actually ask ourselves, shouldn't we aim for more?
I think we should actually. I think, and I would be very. I'm looking. Well, so probably quite a few of us do, right? You know, looking at Eugene Braunwald and inclisiran. You know? Would I take inclisiran injections if they were cheaper? Yeah, I probably would. This way, I take a statin, right? Yeah, but it will be. Yeah, it's an important analysis that we have to do.
So then the European Society of Cardiology, they come up with the very nice guidelines, and I'm trained also as internal medicine specialist. And you have that quite healthy individual with a little bit of higher LDL. And we say, maybe a little bit more relaxed because there's no atherosclerosis yet. Is that the right approach?
I don't think that's the right approach. I think I would treat aggressively early on. So I don't, but you know, so I'm outing myself as very much of an LDL cholesterol believer. I think the slide that I showed with the subclinical atherosclerosis with even lower levels below the treatment paradigm, and look at, well, there's this gender difference. Men get coronary artery disease anyway more often and earlier than women do, and so for men to develop a meaningful or clinically relevant atherosclerotic disease is very, very likely. So having them start early to lower LDL cholesterol, to lower the lifetime burden of LDL cholesterol, I think is a good idea. So that is something I would propagate, and we would recommend patients. It's a bit of an issue of reimbursement in some cases then, but I do recommend.
Yeah, and that also, I think, ties up in your last part of the presentation. Once we are sick, we already have the symptoms. We want to go as low as possible. And we often see that also in cardiology. Isn't it that we have to change our mindset? Once we have a little bit already of advancement, being a little bit sick or not so healthy, to really lower it as much as possible. Of course, maybe the homeostasis of our body cannot go as low as possible. And that's also the reason why it's so hard to go as low as possible.
I think that's the I think we should start when we're not sick.
Yeah.
It's something that we also learned from the new medication, and so that's a very LDL discussion, all right. But what we learned from the PCSK9 inhibitors, we learned how low we can go without having being too low. You know, there was with the statins, there were these FDA warnings on cognitive decline or cognitive disturbances by lowering cholesterol. And the argument could be made, you need the cholesterol for myelin sheath, you need them for the central nervous system. But then again, when you look at PCSK9 inhibitors and the studies, you had these patients with extremely low levels, and they were fine. And you have small children, and you measure in small children, and you have extremely low LDL cholesterol levels. So it seems that the LDL cholesterol is something that builds up in our too affluent society because we're not starving enough in between, and it's something that we can probably from relatively early on start to.
Yeah. Before going to Delphine, because there are many questions also from the audience. My last one, you just said in a half of a sentence, try to implement it into the electronic medical record, didn't work. How are your colleagues looking at you? Are they keen to measure the biological age of their patients? And is the health insurer keen to actually establish a relationship with you because you are really driving your patients into that health mode, lowering the pheno age?
I don't know about the health insurer. I think they would be skeptical because they think I would try to prescribe more drugs that they have to pay for, and they wouldn't see the long-term benefits of it. My colleagues in our medical school, they're very much on board, and the IT people are not the ones that are not totally on board because they haven't implemented it yet. But I hope for, now I can say I hope for 2026.
Okay, so now it's recorded, so that's good. Over to Delphine.
Thank you, Professor Andrea. So thank you, Professor Karsten and Professor Andrea, for the insightful presentation and panel discussion. We have a lot of questions from the floor, so let me begin by asking. First question, in addition to the routine blood-based biomarkers, is the state of gut microbiome also considered among the measures used to measure biological age?
I find gut microbiome very difficult because measuring gut microbiome, I think, is a science in itself, and more of an art than a science. Probably in some cases there have been consortia for that. Right now, we change our gut microbiome whenever we do a colonoscopy prep. And so if we argue, and Andrea, I think we had the discussion 2 or 3 years ago once, or you discussed it and I was listening. Diversity would be sort of something that we cherish as young, and we decrease diversity by colonoscopy prep for a while. We decrease diversity by taking a broad spectrum antibiotic because we have a sinusitis or something. So I haven't seen something that I could implement well and really understand in terms of gut microbiome. So I'm quite a bit of a skeptic there that we can use that very well.
Thank you, Professor. So another question is about omega-3 fatty acid. It was said that it has been linked to atrial fibrillation. So what are your thoughts about this?
Yeah, they have been. Yeah, those data are there. I have a bit of a GI thought about that. That might be totally useless. I come from a background where I did my MD thesis on ventricular arrhythmias and the anti-arrhythmic effect of omega-3 fatty acids. And I saw that effect. I do believe that effect very much. I have co-workers who established epoxy metabolites of omega-3 fatty acids and as antiarrhythmic drugs. So I do believe in the ventricle there's an antiarrhythmic effect. Having said that, all the antiarrhythmic drugs have also pro-arrhythmogenic, or many of them, type 1A to C, etc., of antiarrhythmics have also arrhythmogenic effects. It's hard to argue against the data that are there. The clinical relevance of the atrial fibrillation signal that is seen in those studies is, I think, not a very relevant one. I think there could also be an aspect of when I wrote why I like omega-3 fatty acids so much. I'm not so sure that I like them that much. I'm having here this fish oil thing that I want to take, and I want to I do a sort of little pharmacokinetic study with one of my students here, and I want to take several spoonfuls a day and want to see how quickly the fatty acids go up in my blood. But it's actually quite revolting to take that stuff. And so whenever you take omega-3 fatty acids, and in these studies you take 4 capsules a day, right? You have quite a lot of these fatty acids that you have probably in your stomach. And there's something that is called very nice GI disease, Rumheld syndrome, which is that you have cardiac symptoms from indigestion, because the stomach is really very close to the heart, right? So one of the little sort of pet hypothesis that I entertain in some parts of my of my mind is that maybe this atrial fibrillation effect might be also somewhat linked to the indigestion caused by taking not so nice omega-3 fatty acids. But this kind of far-fetched thought that I'm uttering here, which is now probably now I've said it. I shouldn't probably.
No worries. So another question was about biomarkers. So the phenol-age biomarkers, whether it's measured in men and women, and whether there's any differences between the gender,
Yeah, we have.
particularly for women in the estrogen deficiency, whether there's any differences in biomarker expression.
I don't know. Maybe Andrea can, you know something about that with regard to pheno-age. We haven't looked at pheno-age sort of gender dependent, and I just don't know.
I see. Sure. Another one is whether it perform differently across different ethnicity and what are the ethnic groups that were used in the NINS data set.
That in the enhanced data set, I don't think so. In our data set, it's very, very Caucasian. We're not so ethnically diverse in Brandenburg.
I see.
So in our data set, we couldn't do subgrouping.
All right. One of our audience was also asking whether we could add or substitute markers such as omega-3 or omega-6 indices into phenol-age panel to better capture the aging risk in specific populations.
That's something why I hope I will be able to convince my university to give me a sabbatical next year so that I can come to Singapore to hopefully do that with Jan and Fengsheng, and to take my data with me and together with them and their team to look at how we can use other markers and incorporate them.
Thank you. We'll try to answer. There's really a lot of questions. I'll try to complete it within 5 minutes. We have about 5.5 minutes or so. So from a practical clinical perspective, would you recommend a cholesterol lowering Mediterranean diet as part of a routine strategy to reduce biological age and cardiovascular risk? Because there were some evidences from the PREDIMED trial that showed that Mediterranean diet supplemented with extra virgin olive oil or nuts significantly reduce major cardiovascular events.
Yes, I would very much recommend that. I don't think it lowers LDL cholesterol very much. I think there might be a bit more of an effect on by using the extra virgin olive oil, which is low omega-6. So you kind of relatively increase omega-3s, even though you don't supplement omega-3s very much by taking more olive oil instead of rapeseed or sunflower oil. The Mediterranean diet is not that well defined, but basing the Mediterranean diet on high consumption of olive oil, I think there's strong evidence for that. In terms of looking at nutrition interventions, we have Nutrition Skills Lab here. We have actually measured, you can actually see that small papers coming out from one of my students, that by actually just recommending taking different fats, you can actually lower the omega-6 and increase the omega-3 to omega-6 ratio by just saying use more olive oil. So that's something I think is very, 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 very useful to do. The other thing is in that context is maybe a question also in the discussion space is the thing with intermittent fasting that might be more relevant as compared for LDL cholesterol even than sort of Mediterranean diet, because I don't think you will lower the LDL cholesterol very much by just following a Mediterranean diet.
Thank you, Professor. So another question was, does the link age algorithm also work when some biomarkers are missing? And also, how many biomarkers are required to obtain reliable results?
That is the big, big open question. And that's why I need the sabbatical. And I need to have much more time to spend with Fengsheng and Tango.
Okay. And there was also, I think this is more of maybe a comment. Gavin was saying that regularly donating blood plasma, you visualize how cloudy the plasma is. I think it was just a comment. So maybe, I'll move on.
I just didn't get that. So donating blood to measure something, or to just to sort of
maybe Gavin, if you would want to type in the question and answer again, whether that's a comment or it's a question. I will move on first to the next question. So the next question was it was a comment. I like your hypothesis of connection between gastric events and atrial fibrillation. Interestingly, there is a subspecialty, the gastroenterologist. So yeah, so that's just a comment. And then the next question was, can taking a statin to reduce LDL cholesterol and cutting down on sugar to reduce HbA1c decrease biological age? I think
I would think so, yes.
yes. All right. So for Gavin, maybe we still have about 2 minutes. You could type down in the Q&A while we actually come to the end of the Q&A, but I can continue with a few more questions while we wait for another few more questions if you have. We have about 1 minute and 50 seconds. All right. So Prof. Karsten, I would like to understand also, you were mentioning biological age, and this is a term that we always hear more and more in both research and clinical conversation. So I just want to understand from perspective, what does biological age add beyond chronological age? And why do you think it's becoming more clinically meaningful now?
Because it gives us, from what your research is about, it gives us additional possibilities to assess risk, and also there's this issue in clinical medicine on escalation and de-escalation. When do we escalate therapies? When do we possibly also de-escalate? And I think that biological age can help to steer our intervention strategies. So having a signal of increased biological age might lead us to be more aggressive in treating risk factors, and we don't know. We have to do much more research to understand how we do that. But then again, clinical medicine, a lot of clinical medicine is should be more evidence-based. I think and that's the discussion that Andrea had in the beginning of the Q&A session. How do we use it? I think even though we might not have sort of strong evidence towards one biological clock, we are at a stage where the biological age parameter can inform us to probably change approaches and to define risk differently. Already now.
That's a very nice ending of a very nice webinar. Thank you, Karsten. So keep off on really your great work in translating neuroscience into clinical practice, make it useful and meaningful for all of us. So thank you so much.
Thank you very much for having me, Andrea, Daphne. It was good fun and great pleasure. And it's evening for you now already. I have to go back to work now, but you can hopefully have dinner and we have omega-3 maybe.
We still have a couple of announcements. So the first announcement is that we are preparing the Healthy Longevity intensive course for you. This intensive course is for healthcare professionals. It's happening at the 23rd to the 25th of February. And if you really think, I want to learn more about healthy longevity medicine, or also termed precision medicine, this is the course, 3 days. Then the next 2 days, we have the conference on supplements and repurposed drugs. Really, the only focus is these two topics worldwide, a very unique conference. And of course, the abstract submissions are still open, and we have a startup pitch competition for the conference. And that's happening at the 26th and the 27th of February next year. So thank you for joining. This is our last webinar for a wonderful 2025. At least it was wonderful for the healthy longevity medicine field. I hope you also had a wonderful 2025. We will see each other again on the 15th of January. Professor Thomas Langer is our guest, talking about inflammation and senescence. This will close the 25th of 2025 webinar series with a festive video, wishing you a wonderful holiday season and a very, very happy new year. Take care.
capstone student with the Academy of Healthy Longevity. This is where most of my day unfolds at the clinic. It's a place where science meets real people, the heart of everything.